Greetings to all. In this presentation, you will learn about a multiscale model and its potential to predict soft tissue responses to mechanical loading on both macroscale and microscale. Tricuspid valve is one of the four valves in the heart that regulates blood flow from the right atrium to the right ventricle. The tricuspid valve complex has three leaflets, the anterior, posterior, and septal. There are two types of cells, the endothelial cells, typically found on the surface lining of the valve, and interstitial cells, which are distributed all over the valve. The coordinated action of the tricuspid valve complex is required for a normal tricuspid valve function. Any disruption to these components would result in an abnormal condition, such as the tricuspid valve regurgitation, which is the condition where the three leaflets are unable to coapt completely, causing backflow of blood into the atrium. Currently, this abnormal condition can only be treated surgically by placing a ring or sutures around the annulus. However, a recent study revealed that most of the patients who underwent successful tricuspid valve repair surgeries were diagnosed with mild, moderate, and severe tricuspid valve regurgitation in the long term, irrespective of the surgical procedure. This indicates that the surgical intervention changes the existing mechanical environment of the valve, leading to its gradual deterioration in its function. Therefore, it is critical to understand the effects of surgery on both macro and micro environment of the tissue. Hence, our motivation to develop a computational tool that can provide detailed insight into the changes in the microstructure and microscale biomechanics of the tissue by integrating macroscale response, ECM microstructure, and cellular response of the tissue. Now, to generate a computational model of the tricuspid valve, we characterize the tissue both on macro and micro scale. We extracted a 10 by 10 mm sample from the center of the valve and hooked it to the custom-made biaxial testing device. Tissue response in two directions, that is circumferential and radial directions, were recorded for different boundary conditions. The sample was then unhooked and prepared for scan using the small angle light scattering device. A light source was passed through the sample to get the scattering pattern on the screen, and this scattering pattern was captured and post-processed to get a contour map of the network distribution and fiber direction. The color contour indicates the degree of anisotropy ranging from 0 to 100%. A value close to 0 indicates that the fibers are less aligned, and the value close to 100 indicates that the fibers are more organized or well aligned. The black lines indicate the main fiber direction. Using the network details, we generated a three-dimensional volume element that represents the collagenous and non-collagenous part of the tissue. We generated a three-dimensional finite element model with a mesh size same as the resolution of the SAL scan. The RVEs were then arranged to match the network arrangement in the SALS scan image. The final stress balance equation includes contribution from both collagenous and non-collagenous part of the tissue. Here, the highlighted four parameters, that is the fiber modulus, the stretch ratio, the parameter that controls nonlinearity of the response, and the shear modulus, were used to fit our multiscale model to the experimental data. We simulated biaxial tension test, and as you can see here, our model is able to predict the overall macroscale deformation, including changes in the microstructure. The job took about 20 hours on the supercomputer using 200 processors to solve for over 50 million unknown variables in each time step. Three networks in their undeformed and deformed states from three different regions of the models are presented here. We can observe that the network from the central region remains almost unchanged. However, the networks from the boundary region showed prominent changes in their distribution. That is, they became more organized along the direction of the applied load. In conclusion, to improve the clinical outcomes, it is important to understand tissue biomechanics, and multiscale models can be a tool to provide unprecedented information about the mechanobiology and biomechanics of the tissue, which cannot be obtained from other alternative methods. Our future directions include validating our model, predicted microstructural responses, and include elastin fiber family or intracellular structures.